Another notable effect of hydrogen bonds in water is the observation that water expands as it freezes. To understand this, it's helpful to remember that as molecules heat up, they start to move around a lot. As they cool down, they start to slow down. When water molecules are in a liquid state, they're moving around and kind of jostling about. They're not very organized and their movement is kind of random. As the temperature drops and the water molecules begin to slow down, they settle into positions relative to each other based on their charges. They begin to position their more positive ends towards another molecule's more negative end. Eventually, they form a lattice-like structure. You can also see that as they organize themselves into this lattice-like structure, they take up more space than they did before. On the right is liquid water. The water molecules are able to be closer to each other because they're just moving around. On the left, as they're organized, they actually take up more space. So that means that when water freezes, it actually expands. It also means that ice is buoyant. It floats in liquid water. The hydrogen bonds in water also enable water to participate in many chemical reactions. In dehydration synthesis between glycerol and a fatty acid, you can see that a hydrogen atom and an OH group join together to form a water molecule. In doing so, the glycerol and the fatty acid are now joined. The net loss of a water molecule means that this is considered a dehydration reaction. Remember that these are all effects of the hydrogen bonds in water and the fact that water molecules are slightly polar. In discussing biological reactions, we'll often talk about pH. One way to think of pH value is in the amount of hydrogen ions available in the water. Remember that ions are atoms that have a different number of electrons from the number of protons. When a hydrogen atom loses its electron, it becomes an ion. Solutions with more hydrogen ions, which are also called protons since they're essentially just a single proton, are called acidic. On the pH scale, solutions with more hydrogen ions are lower in value. You can see here that battery acid has a pH of 0. So does hydrochloric acid. These solutions have a lot of free hydrogen ions. Oftentimes, after a water molecule reacts with another molecule, it leaves behind either a hydrogen ion or a hydroxide ion. If the reaction leaves behind more hydroxide ions, there's fewer hydrogen ions relative to the hydroxide ions in the solution. We call this basic. So there's two ways to think of the values on a pH scale. You can think of higher pH values as having fewer hydrogen ions, or you could think of higher pH values as having more hydroxide ions. Conversely, you can think of lower pH values as having more hydrogen ions, or you can think of a lower pH value as having fewer hydroxide ions. This can be a little confusing. The easiest way to remember it is that lower values are more acidic, higher pH values are more basic. Many homeostatic processes in your body are devoted to maintaining pH within different systems. Now that we've talked about atoms and the ways that atoms can interact with each other, let's talk about 4 major macromolecules. The theme of macromolecules will occur repeatedly during the semester, so it's something that you really want to get a hang of. The first macromolecule we'll talk about are carbohydrates. Carbohydrates include simple sugars and complex carbs. Simple sugars are mono and disaccharides, while complex carbs are polymers. They're big chains of monosaccharides. Simple sugars serve to provide quick energy, whereas complex carbs often serve to support cells or store energy more long term. You're probably familiar with table sugar, which is a simple sugar, but cellulose is also a sugar. Cellulose is the carbohydrate that makes cell walls in plants. So when you see a big tall tree, most of the wood and vegetative material is actually a kind of complex carb. The carbon is derived from the air. So when you see a big tree or a plant that's made up of a lot of different vegetative structures, you're actually seeing the product of that plant's sequestering of carbon from gases in the air. That mass came from the air all around. Here's a monosaccharide, which is a simple sugar that is available naturally in things like fruits. We often consume disaccharides in the form of table sugar. Polysaccharides are long chains, polymers, of monosaccharides and disaccharides. These include structural things like the exoskeleton of an insect. The hard outer shell of an ant is actually made out of a carb, or a sugar. The next major macromolecule is protein. Proteins are polymers of amino acids. Remember that polymers are chains of smaller units. Proteins carry out cellular work. Polypeptides are strings of amino acids that make up larger proteins. You can see here that this polypeptide is made out of 6 different amino acids. We'll discuss later how amino acids are made in the body, but it's helpful to notice that this is another example of smaller structures making larger structures. These amino acids form a chain called a polypeptide, and several polypeptides will form what's known as a protein. Here you can see that strings of polypeptides are forming a shape. These shapes sometimes serve a function, and we call these functional proteins. Different areas in this cluster of polypeptides, or functional protein, have different ways of interacting with other molecules. An enzyme is a kind of protein that speeds up a molecular interaction. So, in this example on the top, that dark gray area is the enzyme. The molecules that interact with the enzyme are called substrates. Starting from the left, you can see that the enzyme approaches the substrate and settles into an area that has the right shape for that substrate. Once it connects, some kind of interaction occurs, and the enzyme is able to break that substrate apart. It then releases the substrate, and the end result is what's known as products. The product is just the end result of that enzymatic reaction. The reason why enzymes are important is because they lower the amount of energy required in order to make some reactions occur. Without enzymes, those reactions could still occur, but they would take a lot of energy input, either in the form of heat or the accumulation of heat over time. With enzymes, reactions can occur very rapidly. Here there are 2 lines, a blue line and a red line. These lines illustrate the energy needed to convert CO2 and water into carbonic acid. On the y-axis is energy. So the higher these lines go means the more energy is used. You can see that with an enzyme present, the blue line ends up using less energy in order to achieve the same amount of product. Without an enzyme, which is the red line, you can see that much more energy is needed in order to gain the same amount of product. Here's an example of a conversion of proteins. You're probably familiar with this process, but now let's talk about what's happening. Here's an uncooked egg on the left. As we heat the egg, the proteins become opaque, and finally they become insoluble. What's the difference? Well, if you've ever put egg white in a drink and shaken it up, you end up with a mixture, kind of like a foam. If you ever tried that same thing with cooked egg whites, you won't get that same effect. But what's the difference? In their natural state, these functional proteins associate freely in their original structure. As they're heated, the energy in the system starts to unravel, unwind, and disassociate those proteins from each other. Finally, with enough heat, new interactions occur between the polypeptide strands, and we have what's known as cross-linking. This is kind of like the disorganized reassembly of those proteins. We call this process denaturation. Another way of saying it is that the egg is cooked. Denatured proteins no longer function. Heat is one way that you can achieve denaturation, but there are other processes that can lead to cross-linking. Can you think of another way that we cook foods without using heat? Ceviche is a dish that uses another process to denature proteins. It uses an acidic solution, usually a mixture of lime and different citrus fruits, in order to denature the proteins on the outside of pieces of fish. This is often called chemically cooking the fish. It serves as another pathway to denaturing and cross-linking the proteins that doesn't use heat. Another kind of macromolecule is a nucleic acid. Nucleic acids are also polymers of nucleotides. Nucleic acids are often used to store genetic information. Nucleic acids consist of a phosphate group, a sugar, and a nitrogen base. Ribose is the sugar in RNA, whereas deoxyribose is the sugar in DNA. There are several different nitrogen bases. Cytosine, uracil, thymine, guanine, and adenine are all nitrogenous bases found in nucleic acids. Uracil is only found in RNA, whereas thymine is only found in DNA. In this illustration, you can see the interaction between 4 nucleotides. Since we have thymine on the top left, we know that these are components of DNA. An important feature in the interaction between nucleotides are the hydrogen bonds. It's helpful here to note the number of hydrogen bonds that are occurring between the different nucleotides. You can see that the thymine group and the adenine group are both associating by 2 hydrogen bonds. This is different between the guanine and the cytosine groups. These are associating between 3 hydrogen bonds. This is really helpful to know. These nucleotides only are compatible with one other kind of nucleotide. This means that if you encounter a thymine, you know that the only other nucleotide that could associate with it is an adenine. Conversely, if you find adenine, you know that the only complementary nucleotide is thymine. Further, if you find guanine, the only nucleotide that will connect with 3 hydrogen bonds would be cytosine. Lastly, if you have cytosine, you know that the only nucleotide that interacts with cytosine is guanine. Stacks of nucleic acids form a familiar double helix shape. What's important to note here is that you only really need the information on one side to know what that complementary strand would look like. The last major macromolecule that we'll discuss are lipids. The structure of triglycerides includes glycerol and 3 fatty acids. Triglycerides are used in the storage of energy. Phospholipids contain glycerol and 2 fatty acids, and these often form biological membranes. Some lipids form steroids. Structurally, steroids have 4 fused rings. Steroids function to stabilize an animal's membranes, and they also serve as hormones. Examples include sex hormones like estrogen and testosterone. On the left is an example of a saturated fat. Saturated fats are kinds of lipids that have 2 hydrogen atoms for every carbon atom. This leads to straight carbon chains. On the right is an unsaturated fat. Some of these carbon atoms have double bonded and only have a single hydrogen atom. That leads to a kink in their structure. Saturated fats include things like butter. Unsaturated fats often include things like oil that are liquid at room temperature. Steroids are biologically active forms of lipids. All steroids have 4 carbon rings. Here you can see that the 4 rings are represented by the letters A, B, C, and D on the top left. Each bend in these rings represents a carbon atom. You can see the similar structure, just like A, B, C, and D, in all of the other examples of steroids. Lipids form a group of very important biological structures. Energy storage in the form of fats, biological membranes like the membranes that surround cells, and hormones are all very important in maintaining homeostatic processes in animals.